Video is ended. Video streaming
Sorry for the interruption, students. Once again, see the type of sex determination in case of human beings. The type of sex determination in case of human beings is determination. Males are heterogametic. Males produces two types of sperms. Okay. So see here, males produces two types of sperms. Okay, heterogametic. They produces 50% of the sperms carry X chromosome. So those sperms which are carrying the X chromosome, they are known as gymnosperms. They are known as gymnosperms. And those sperms which carry and remaining 50% of the sperms carry Y chromosome. Y chromosome. That these Y carrying chromosome, they are known as androsperms. They are known as androsperms. Okay. This Y chromosome was discovered by Stevens and Wilson. Stevens and Wilson. And they said that Y chromosome, chromosomal basis of sex determination in case of human beings, this Y chromosome is essential for presence and absence of Y chromosome is determining the sex in case of human beings. Okay, what they have told that Y chromosome, presence and absence of Y chromosome determines the sex. In humans. Okay. According to chromosomal theory of sex determination by Stevens and Wilson. Okay. So those sperms carrying X chromosome, they are gymnosperms, and those chromosomes carrying Y chromosome are androsperms. Okay. So in case of male heterogamity, who will determine the sex of the offspring? Fertilizing sperm will determine the sex of the offspring. So see here. XX, XY. Females produces same type of ovum, whereas male produces two types of sperms with X and Y. So if the ovum is fertilized with the sperm carrying X chromosome, then it is female. If the ovum fertilized with the sperm carrying Y chromosome, then the sex of the offspring will be male. Okay, so in case of male heterogamity, sex of the offspring will be determined by the fertilizing sperm. And the next method is, the second method is XX-XO method. Okay. So XX-XO method of sex determination can be seen in case of grasshopper, certain bugs, and cockroach. Grasshoppers and certain insects and cockroaches, the type of sex determination is XX-XO method. Okay, so all the females produces same type of ovum which carry X chromosome, whereas males produces two types of sperms. In that 50% of sperms they carry X chromosome, and remaining 50% of the sperm that do not have X chromosome. Okay, so based on this, if the ovum fertilized with the sperm carrying X chromosome, it is female. If it is fertilized with the sperm which does not carry X chromosome, so it is AAXO, then it is male. Okay. So this type of sex determination is known as XXXO method of sex determination. The next method of sex determination is ZZZO method of sex determination. That is female heterogamity. It can be seen in case of Humia moth and in other insects like butterfly. See the third method, ZZZO method, which is seen in case of Humia moth and butterfly. Okay, and many insects, other insects also. Here, okay, males are homogametic, they will produce only one type of sperm carrying Z allosome. Okay, whereas heterogametic, they will produce two types of ova. 50% of ovum carry Z chromosome and remaining 50 does not carry this Z chromosome or Z allosome. Then it will be X. ZZ, well it will be developed into male, and ZO is going to be developed into female. It's going to develop into female. Okay, it is female heterogamity. Okay, this type of sex determination is female heterogamity. Another example for female heterogamity is ZWZZ. Okay, these are females, these are males. And females, it is producing two different types of ova. 50% ovum they carry Z allosome, and 50% of ovum they carry W allosome. Whereas all the males produce only one type of sperm, they carry one Z chromosome. Okay. Now the sex of the offspring will be determined by the fertilizing ovum. Okay. So this is male and here female. And here it is female. So in case of female heterogamity, sex of the offspring is determined by the fertilizing ovum. Okay, the fertilizing ovum will determine the sex of the offspring because two different types of ova are produced in both the cases. Okay, so this is heterogamity, male heterogamity and female heterogamity, chromosomal method of sex determination. Okay, and the next method is, before going to the genic balance theory, what is mentioned in NCERT is haplodiploidy. So haplodiploidic method can be seen in case of honeybees and many Hymenoptera insects. Haplodiploidic method of sex determination. So see here, this haplodiploidic method of sex determination can be seen in case of Hymenoptera insects. Mostly in case of honeybees, certain wasps. Okay, these types of sex determination. What is this haplodiploidic method? So see here, males are in case of honeybees, that is Apis indica or Apis mellifera, scientific name of honeybee. See, males are haploid where n is equal to 16 chromosomes. Okay, and females are diploid. Females are diploid where 2n is equal to 32. See, in case of human beings and in case of Drosophila, both males and females are having equal number of chromosomes. Do remember this? Human beings, birds and reptiles, all they are having this. I forgot to tell you. ZWZZ method of sex determination can be seen in case of fishes, reptiles and birds. ZWZZ method of sex determination is seen in case of fishes, reptiles, birds. Okay, and now see here, males and in all those insects and even in case of human beings and Drosophila, the number of chromosomes in both males and females is equal. Whereas in case of these honeybees, males and females are not having equal number of chromosomes. Males are having haploid set of chromosomes, whereas females are having diploid set of chromosomes. N is equal to 16 in males and 2n is equal to 32 in case of females in honeybees. Okay. Now, see the method of sex determination in honeybees. So a female with 32 chromosomes during oogenesis undergoes meiosis. During oogenesis undergoes meiosis and produces ovum. Here 2n is equal to 32 and produces n is equal to 16, n is equal to 16 ovum. Okay. Those ovum which are fertilized, whereas in case of males, where n is equal to 16, during spermatogenesis they will not undergo meiosis but they undergo mitosis. So they will undergo mitosis. Spermatogenesis. Okay, and produces n is equal to 16 sperms. Okay, n is equal to 16 sperms. Then, if the sperm is going to be fertilized with the ovum, if the ovum fertilized with the sperm, then the resultant zygote 2n is equal to 32. And we know that if the organism is having 32 number of chromosomes, it is going to be developed into female. So these are females. These are going to be developed into females when during fertilization. Okay. So when fertilization occurs, it results in female sex. Okay. And those ovum which are remain unfertilized, the unfertilized ovum is directly developed into males. The unfertilized ovum unfertilized. Development of any organism without fertilization or development of a new offspring without fertilization, that process is known as parthenogenesis. Okay, it is going to develop into n is equal to 16 sperms. It is going to develop into males. Okay, and the parthenogenesis, which involves in production of only male individuals. If the unfertilized ovum, see, there are two chances. It can be developed into female or it can be developed into male. But the unfertilized ovum developing only into male, such type of parthenogenesis is known as archenotochy. Okay, so the type of parthenogenesis seen in case of honeybees is known as archenotochy. Okay, so what is this? By parthenogenesis. That it is known as archenotochy. And it is known as archenotochy. So this male has no father. By the ovum which is unfertilized is developed into male. So there is no father for this honeybee. Okay, no father. And next, and the same male, it is again undergoing mitosis and produces sperms. N is equal to 16. Okay, this is going to be, if it is going to fertilize by the ovum, N is equal to 16 by the ovum, then these are going to develop into, so n 2n is equal to 32. Again the sex of this is female. Then the sex of this is female. So the so-called male, they are having daughters but there are no sons. Okay. When this daughter participates in oogenesis, and by a process called meiosis, and produces two different types. Produces ovum, n is equal to 16, n is equal to 16. If the unfertilized ovum, it can be developed into sperm. Unfertilized ovum, it can be developed into male. Okay, that means the males which are produced, they do not possess father and they do not possess even sons, no son. But the same males, they have grandfather and grandsons. These will be grandsons. Okay. This type of sex determination, it is known as haplodiploidic method of sex determination. And if you see in case of a honeybee population, there are three different populations in honeybees. What are they? In the honeybee population, where 2n is equal to 32, one which is fed with royal jelly, is going to develop into fertile queen. It is going to be developed into fertile queen. Okay. And the other bees which are feeded with pollen and nectar, they are going to be developed into worker bees. Worker bees. Okay. These all are females. These all are females. But these worker bees are sterile. And males, all males, n is equal to 16. They are going to be developed into males, and these males are called drones. These males are called drones. Okay, so there are three sets of population. One is queen bee, worker bees, and drones. Okay, this method of sex determination, which is seen in case of honeybees, is known as haplodiploidic method of sex determination. And the other method, it is genic balance theory, which was proposed by Bridges. See the next method. Up to now we have seen two methods. One method is chromosomal method of sex determination in that XXXY, XXXO, where it is male heterogamity. And the other one is female heterogamity, that is ZOZZ or ZWZZ, that is female heterogamity, that is chromosomal method of sex determination. And the second method what we have seen, it is haplodiploidic method of sex determination seen in case of honeybees. And the third method of sex determination, it is genic balance theory. See, genic balance theory. This genic balance theory was proposed by Bridges. And this genic balance theory, according to Bridges, he is stating that sex of an offspring or sex of an individual will be determined by the ratio of X chromosomes to the number of autosome sets. Once again I repeat, according to genic balance theory, which is given by Bridges, determined by the ratio between number of X chromosomes to number of autosome sets. Is that ratio, it is known as sex index. Okay, hope I am clear. Once again see here. According to this genic balance theory, the sex of an individual is determined by the ratio of X chromosomes, number of X chromosomes by total number of autosomal sets. Okay, this ratio, it is known as sex index. This is known as sex index. Once again see here, the sex of an offspring is determined by the number of X chromosomes divided by total number of autosome sets. Okay, so here this is known as sex index. Now, if the based on the karyotypes, if XXAA, then the ratio is XXAA 1/1, that is 1. Okay, the sex ratio is 1. Then the offspring is female. Okay. If it is XYAA, then the sex ratio is 0.5. Then the sex ratio is 0.5. Then the sex of an individual is male. And anything which is above 1.0, then she will be meta female. If between below 0.5, it will be meta male. If it is in between 0.5 and 1.0, then it will be intersex. Okay. So see here XXXO AA. So here 1.0 again. Okay. Sorry, it is 0.5 male. But see here, Y chromosome is essential for determining the maleness. Okay, so these males are fertile males who are having Y chromosome. But here it is lacking Y chromosome. But Y chromosome is essential for determining the fertility of the male. Because of absence of Y chromosome, it has not turned to be female. But because of presence of Y, it can turn to be male. But Y chromosome determines the fertility factor. So it is a sterile male.